Done?
Done.
Right. Right and left. What?
Okay.
Just check with her if you
Yeah. Sorry for the delay. This class on carbohydrates has been postponed. Finally, we are there. So let us see. We have talked about the molecules, and I will cover the carbohydrates. So first, let's come to what are carbohydrates. We know that these are macromolecules that are produced by plants, and humans eat the carbohydrates and they convert it into glucose to give the energy. And glucose is the most abundant carbohydrate, and it is the preferred source of energy for the blood, brain, and the nervous system. And if you look at the diet pattern globally, you'll find that carbohydrate rich plant foods make up the foundation of all the diets, irrespective of the country that we are in. So, carbohydrates, they are organic compounds which are made up of carbon, hydrogen, and oxygen. They are, and they are aldehydes or ketones or their derivatives. Commonly, if you look at carbohydrates, the hydrogen and oxygen ratio in the carbohydrates is very similar is to 1, and therefore the name came carbohydrate, carbon, and hydrate. So actually, carbohydrates are hydrates of carbon. So sometimes carbohydrates, we refer to them as saccharides or simply as sugars. And glucose is the simplest one, and it has a formula of C6H12O6. Most of the carbohydrates are named and they have an OSE at the end. Example, glucose, lactose, which is milk sugar, maltose, which is malt sugar, and fructose, which is fruit sugar. In animals, the main sugar is glucose. It is converted into energy, but the storage form of glucose is glycogen. In the plants, there are a wide variety of monosaccharides and oligosaccharides. However, the storage carbohydrate is starch. And then you have the polysaccharide cellulose, which is the structural polysaccharide. It is found in the cell wall of all plants. The gums also are a group of polysaccharides which are obtained from plants and seaweeds, and we will see what these gums are later. If you look at the principal carbohydrates in the human diet, this is in the diet of humans. This is in the diet of humans. We can divide them into polysaccharides, dextrins, and free sugars. The disaccharides, the disaccharides, the oligosaccharides, and the sugar alcohols, which are products of these saccharides. Under the polysaccharides, we can divide them into starch and non-starch polysaccharides. These non-starch polysaccharides are the polysaccharides which are known as dietary fiber. In starch, we have amylose and amylopectin, and the non-starch polysaccharides include cellulose, hemicelluloses, pectins, the xylans, and the gums. So the dietary fiber portion of a carbohydrate are the non-starchy polysaccharides or non-starch polysaccharides. So we also find polysaccharides not only in the plants or anything, they are also very important in the human body. You will find that the number of carbohydrates are formed on the surface of cells. They act as road signs road signs distinguish one cell from the other. In fact, all your blood group marking, that is, the blood group markers, are designated as A, B, and O or AB plus, depending on the carbohydrate which is there on the cell membrane. Heparin, which is the anti-clotting factor in the blood, also is a carbohydrate. Chondroitin sulfate, which you find in your joints, also is a carbohydrate. So let us see the classification of carbohydrates. Carbohydrates can be classified by different ways. The first and the most simple classification of carbohydrates is as monosaccharides. So based on the number of carbohydrate units or the sugar units. So with a single sugar unit, it is a monosaccharide like glucose and fructose. If there are two sugar units, it is known as a disaccharide, sucrose, lactose, as well as maltose. Then you have oligosaccharides, which are there, which contain anywhere between 3 to 9 residues. Examples are raffinose, stachyose, verbascose. These are oligosaccharides which are found in the plant but not digested by the human because we lack the enzyme alpha 1,4 galactosidase. Polysaccharides contain saccharide units or sugar units 10 or larger, and this includes cellulose, glycogen, starch, amylose, amylopectin. So, based on the number of carbohydrate units, we have four different classes. Then we can classify them based on their functional group. So monosaccharides which have got an aldehyde group on the carbon 1 are called aldoses. So any monosaccharide which has an aldehyde group is known as an aldose. And then any one which has a keto group on the C2, which we are seeing, what I'm showing you is on C2, that is the keto group, C double bond O, it is a ketose. So you have aldoses and you have ketosis, depending on whether they have the aldehyde group or they have the keto group. Then you can classify them based on the number of carbons. The simplest carbohydrate or the simplest aldose, which is a 3-carbon sugar known as triose. You have glyceraldehyde, which has 3 carbon and it has an aldehyde group. And the keto form of glyceraldehyde, the keto counterpart of glyceraldehyde is dihydroxyacetone phosphate. If it is 4 carbon, it is tetrose, 5 ribose, hexose, heptose, heptulose, etc. So now we have the aldoses, the aldohexose, that means it is a 6-carbon sugar with it is a 6-carbon sugar uh, with a aldehyde group, and we have the ketopentose, that means it is a 5-carbon sugar with the keto group. Uh, Now, if we look at the structure of carbohydrates in 3 different ways, they are projected in 3 different ways. One is the Fischer projection, which is on the right. Then you have the Haworth cyclic structure. And third is the conformational formula, which is either in the chair form or in the boat form. Let us look at these three configurations in detail. So if you look at the Fischer projection, if we look at the Fischer projection, we write it with the hydroxyl groups, and we have the first carbon on top and the last carbon at the bottom. So this is the Fischer projection where the hydroxyl groups are either on the left or on the right. And conventionally, we write it like this. But when writing reactions, it is very difficult to keep writing such big formulas. So we have in the Fischer projection can be written as cyclic hemiacetal forms of D-glucose. So aldohexoses or ketohexoses of the sugars in solution will undergo a cyclization and they form a hemiacetal or a hemiketal in the aldose or ketose. If you look at the structure of glucose, it contains an aldehyde group and it contains 5 hydroxyl groups, either projecting to the left or projecting to the right. Now, in solution, when you have, there can be an intramolecular interaction and a rearrangement. So here, the carbonyl group, that means the aldehyde group on C1, will interact with the carbonyl 5, that is the number 5, what is written on the right, and it will produce a closed ring, which is called as a Pyrenees ring. So the reaction results in a ring which is termed as the cyclic hemiacetal. That is, it links the carbonyl carbon with the carbon 5 of the sugar. So it becomes, so what happens to the carbon 1, which is there, it becomes asymmetric, resulting in have having the hydroxyl group to the right of C1 is designated as alpha D glucose. That is what you can see in pink. And if that hydroxyl group is on the left of the C1, then it is known as beta D glucose. So we have alpha glucose and we have beta glucose. These two forms of glucose are known as anomers. That is, anomers are sugars which differ in the configuration around C1. So this is referred to as the anomeric carbon, and you need to know about the anomeric carbon because this anomeric carbon actually tells us whether the sugar will be a reducing sugar or it will be a non-reducing sugar. So the second structural way of writing the sugars is because in the Fischer projection, it is not very easy to write the true shape of a cyclic hemiacetal. So Haworth suggested a new formulation in which the ring can be shown as a planar hexagon. So in the Haworth cyclic structure, the 6-membered ring is shown. The 6-membered ring is shown as a cyclic ring with an oxygen, and it has got its name from, and these are called as pyranoses because A six-membered ring with an oxygen in organic chemistry is known as a pyran. It is a heterocyclic compound. So any carbohydrate which we write in the six-membered ring form is known as a pyranose, and the glycosides of these are known as pyranosides. Now, the alpha form of the glucose you will see is when the hydroxyl group is opposite to the CH2OH, which I have circled over here so that it is visible. And in beta glucose, the CH2OH and the OH will be on the same side. So you have alpha D glucopyranose and you have beta D glucopyranose. You will see similar structures. You can write similar structures with alpha D galactose and beta D galactose also. Now coming to the keto sugars, which have these in the Haworth projection. These can be written as five-membered rings where you have the C2. Remember that here it is the carbonyl carbon is on C2, whereas in the aldohexoses it is on C1. So the C2, which is shown in blue, will react with the OH, which is shown in pink, and you have a cyclization. And here, instead of a six-membered ring, you get a five-membered ring, which is called as a furan. And this is a furanose. And sugars which have this five-membered structure are known as furanosides. So we have the pyranosides and we have the furanosides. So if you look at alpha fructofuranose, when the OH group is on the opposite side of the CH2OH, it is an alpha anomer, and when it is on the same side, which I have circled there to make it visible, then it is beta D fructofuranose. So alpha D fructofuranose and beta D fructofuranose are anomers. Now, it is known that in the sugars, even though the Haworth structure is supposed to look like a planar hexagon, it is not planar, and it was found that it does not represent the correct conformation of the monosaccharide, and the monosaccharides are now represented in two different conformations called the chair form and the boat form. The conformational structure in the chair form is more stable than the boat form. So if you look at the structures, you have alpha D glucopyranose written in the chair and in the boat, and you have the hydroxyl group circled to tell you which of this is on the opposite side or same side as that of CH2OH. So we can write the structure of carbohydrates either in the Fisher projection or in the Haworth projection or in the boat and chair conformation. So what is it that makes these anomers? It is very important for you to know about and identify which the anomeric carbon is when you write it in any one of these projections. Because when you're looking at whether a sugar is a reducing sugar or a non-reducing sugar, the anomeric carbon must be free. It must not be in linkage with another sugar. And we will see later how the anomeric carbon tells you whether it is a reducing sugar or a non-reducing sugar. So here is the projection of erythros to show you the anomers of erythros. So you have D-erythros on the left and the beta D erythros on the left and alpha D erythros on the right, and you have the Fisher projection on the extreme left. Then in solution, these anomers, alpha and beta anomers, are in equilibrium and they keep converting between the alpha to the beta form. Even if you purify the anomer and you crystallize it, when you put it into solution, part of it will become the beta anomer. So this type of equilibrium or conversion between the alpha and the beta anomers is known as mutarotation. Then let us come to the isomerism in carbohydrates, which we are very famous for. What is an isomer? An isomer is a compound which has the same molecular formula and two different structural formula of functional molecules. The word isomerism comes from the Greek words isos meaning equal and meros meaning part or to share. So glucose and fructose are structural isomers. Glucose and fructose are structural isomers. Glucose and glucose and the other pyrenoxides are structural isomers. Galactose and fructose are also structural isomers. Then we have stereoisomerism. This comes from the word stereos meaning solid, and this is the way the molecules are in space, distributed in space. And the stereoisomerism is the spatial arrangement of the OH and the H. And then we have optical isomers. So in stereochemistry of the monosaccharides, what you need or what you need to understand is that there should be a chiral carbon. So what is a chiral carbon? A chiral carbon is a carbon which is attached to four different atoms. Only if it is attached to four different atoms, it can be known as the chiral carbon, and it can exist as either the D form or the L form. So if you look at the figure there, you see that if the center represents the carbon, the horizontal line is coming out of the plane of the page towards you. So we generally put it in dark, what you can see on the extreme right. And the two groups which are on the vertical line go behind the plane of the paper, that is away from you, and so therefore we generally put it as hashed line. So look at the structure of glyceraldehyde. You have a chiral carbon which is attached to four different groups: a CH2OH group, a OH group, a H group, and an aldehyde group. So that carbon is a chiral carbon, and it can exist as L-glyceraldehyde, where the OH is on the left-hand side, which is LEVO, or you can have D-glyceraldehyde, where the OH group is on the right-hand side, and it is called as the dextro. So you have the capital D and the capital L notation. This is an old notation, but for carbohydrates and for amino acids, we still continue to use the D and L notation. We have, instead of the D and the L notation, we have the R and the S. In most organic chemistry books, you will find the R and the S notation. So in stereoisomers, now let us look at the glucose. If you see numbered chiral carbon here, in arabinose it is carbon 4, in glucose it is carbon 5. So you have D-glucose and you have L-glucose, and the D and the L are designated according to the highest numbered chiral carbon. Okay, so If in the highest numbered chiral carbon, the OH is to the right, it is known as D, and if it is to the left, it is known as L. Similarly, look at L-arabinose also. The highest carbon numbered chiral carbon is carbon 4, so it is to the left. So D in Latin is dextro, and in the present thing it is R, which is rectus, Latin for right, and you have L, which is levo, and S. R and S are the current notations for the stereoisomerism. But as I said, we still use D and L for sugars and amino acids. And most naturally occurring carbohydrates are all of the D configuration. This is exactly opposite that of amino acids. All naturally occurring amino acids are L amino acids. So remember that the sugars are the D sugars and the amino acids are the L amino acids. This is the capital D and the capital L. So to tell you again, D-glyceraldehyde, highest carbons. Look at all the highest carbons. So L. And these two types of stereoisomers, which have the same formula and they differ in the configuration of one carbon of the D and the L, they are known as enantiomers. So the L and the D is a means of describing the configuration of the sugars based on the chiral carbon. So the D-glucose and L-glucose are enantiomers and L-alanine and D-nitrosis. Let us look at the chiral carbon and see how many stereoisomers do exist for each of these sugars. So if you look at the glyceraldehyde, you have only D or L. That means there are two. If you look at erythrose, which is a 4-carbon sugar, you have got 4 stereoisomers: D-erythrose, L-erythrose, D-threose, and L-threose. Now, if you go to ribose, which has 5 carbon numbers, it is an aldopentose. If you look at the number of the isomers, you find that it has 8 isomers. And if you look at the C6 sugars, which have got 4 chiral carbon, they have got 16 stereoisomers. So the formula for finding out the number of stereoisomers in the sugars is the 2 to the power of the number of chiral carbon. So in aldopentose, you have 3 chiral carbons, so you have 8 isomers. In aldohexose, you have 4 chiral carbons, so therefore you have 16 stereoisomers. So glucose can exist as 16 stereoisomers, of which 8 of them are D and 8 of them are L. So you have D-allose, D-altrose, D-glucose, D-mannose, D-gulose, D-idose, D-galactose, and D-talose. Of these, the more common are D-glucose, D-mannose, and D-galactose. Now, optical isomerism. Here, a compound is said to have optical activity if it rotates plane polarized light passing through it. The two compounds having a similar formula will have different optical activity. If they rotate the activity, the plane polarized light in the clockwise direction, they are known as dextrorotatory or with a small d. Remember, optical isomers are written with a lowercase d and lowercase l, whereas the stereoisomers are written with a capital D and a capital L. So when it rotates the plane polarized light to the left, it is known as the L isomer or the minus isomer. So what you see on the right is the current notation where it is given as R, which represents the stereoisomer, plus what is written in parentheses is the optical isomer of glyceraldehyde. So you have R plus glyceraldehyde or D plus glyceraldehyde, and S is L minus glyceraldehyde. Now, what are look at them? Your left hand is the exact mirror image of the right hand. So your left hand and right hand are enantiomers. So if you look at D glucose and if you look at L glucose and you look at it in the mirror, they are exact mirror images. So the capital D and the capital L forms of glucose are known as enantiomers. So stereomers, which are mirror images of each other, are known as enantiomers. So stereoisomers that are not enantiomers are known as diastereomers. So if you look at D-glucose, the stereoisomers of a hexose, you find that there are 16 different stereoisomers. So among them, D-glucose and L-glucose are stereoisomers. The other 14 are all diastereomers of glucose. So D-allose, altrose, glucose, mannose, gulose, idose, galactose, these are all diastereomers of D-glucose. So here is it to again simply tell you that D-glucose and L-glucose are L-enantiomers, and those which have the same formula as that and which are not enantiomers are diastereoisomers. We have one more type of isomerism in sugars differ only by one chiral center. So look at the structure of D-glucose and D-galactose. They are almost identical except for carbon 4. So if you see in carbon 4, the OH for glucose is on the right hand side, whereas for galactose is on the left hand side. So that chiral, that is the single chiral carbon, there is here you have the chiral center is differing. So D-glucose is an D-glucose and D-galactose are epimers. So D-galactose is an epimer and is also a diastereomer. Look at mannose, which is on the left hand side. You look at the chiral carbon 2. So here again, it only differs in that. So therefore, D-mannose is a C2 epimer of D-glucose. And then we have a group of sugars where the oxygen, the hydroxyl is removed and you have the oxygen there. And this we call as deoxy sugars. You must recollect that we had looked at deoxy sugars when we talked about deoxyribonucleic acid. So 2-deoxy-D ribose. If you look at the second carbon, which is shown in green over there, you see the OH, the oxygen has been removed and you have a hydrogen. So it is known as deoxyribose. And then you have amino sugars where a hydroxyl group can be replaced by an NH2, an amino group, and the hydrogen of the amino group can be acetylated and you can have what is known as N-acetyl D-glucosamine or N-acetyl muramic acid. So nutritionally, important monosaccharides, glucose. Blood levels are very important, especially for diabetes mellitus. Is the most abundant monosaccharide. It is a part of almost every disaccharide, and it is the only monosaccharide which is present in starches. Fructose is the fruit sugar, sweetest of the monosaccharides, and it is an epimer of glucose. Galactose is a part of the disaccharide lactose. It has a single chiral center, and it is a C2 epimer of glucose. Mannose is found in cranberries and is a C4 epimer. So this is to just show you the structural differences so that you understand them between glucose, galactose, and fructose, which are all structural isomers. Then you have the disaccharides, which are made up of two sugars, and when they are hydrolyzed, either or broken down by either an enzyme or by dilute acid, they will give the constituent monosaccharide. So they will give two sugars, and so they are known as disaccharides. So each of sugars is linked by a linkage, which we call as a glycosidic linkage, depending on which carbon is involved in them. So when two glucose molecules unite with the loss of water, they form maltose. And if you look at the linkage, it is an alpha 1 4 linkage. So it is the anomeric carbon of one glucose which forms a linkage with the carbon of the second glucose. But remember that the anomeric carbon of the second molecule is free, and so therefore maltose is a reducing sugar. If you look at glucose and fructose, the sucrose, it is made up of glucose and fructose, and here you have a 1,6 linkage, and in this case the anomeric carbon is involved, and therefore sucrose is a non-reducing sugar. And lactose is made up of galactose and glucose. So maltose, as I explained, is made up of two glucose molecules. It is also known as the maltose sugar. So it comes from the partial hydrolysis of starch, and it is used in cereals, candies, etc. Now just let us look at how maltose is formed. So what is shown in pink is the linkage on the left hand side with one carbon 1, which is the anomeric carbon of alpha D glucose, and it links with the 4 of the second glucose. So it is linking alpha 1 4, and if you see that the OH group is on down, the alpha anomer, the anomeric carbon is free, so therefore it is a reducing sugar. Lactose is known as the milk sugar, and it is made up of beta D galactose and alpha D glucose. So you see beta D galactose, the OH is on top. So carbon 1 is the anomeric beta, and it forms a glycosidic linkage with the OH of alpha D glucose. So therefore, this linkage is known as a beta 1 4 linkage. Once again, look at the right hand side, the alpha D glucose molecule. The anomeric carbon is still free over there, that is a, so therefore lactose also is a reducing sugar. Lactose can be hydrolyzed by the enzyme beta galactosidase. It can break down lactose into D-galactose and D-glucose. Many people develop lactose intolerance, maybe due to the antibiotics and also due to allergy reactions, and milk sugar cannot be digested. It results in flatulence, in cramps, and diarrhea. And we have also seen that it is an inborn error of metabolism of some children who have a defect in the gene and who are um, lactose intolerance from day one of their birth. Sucrose is the most common disaccharide, that is the sugar. Structure of it, it is that of the glucose with fructose, and I have shown the anomeric carbon with a star. So the anomeric carbons are involved in this 1,6 linkage. So therefore, sucrose is a non-reducing sugar, and sucrose is hydrolyzed in the human gut by the enzyme sucrase, which is secreted in the small intestine, and these monosaccharides are absorbed directly into the bloodstream. Invert sugar is something which you hear of often in food technology. It is a mixture of glucose and fructose, which is prepared by the hydrolysis of the sucrose, either by acid or enzyme. The enzyme is invertase, which breaks down sucrose into glucose and fructose. And when sucrose is broken down, you have equal quantities of glucose and fructose, and therefore it is known as invert sugar. Now, invert sugar has got a low water activity compared to sucrose, and so therefore it is used and it is preferred by food manufacturers for the preparation of packaged foods. And invert sugar also retards the crystallization and it retains the moisture of the packaged food. It acts as a humectant, that means keeps the water inside. It acts as a water binder and it promotes non-enzymatic browning, widely used in the food industry. So invert sugar is the hydrolysis product of sucrose. Then you come to the oligosaccharides, which have more than two sugar units, and the common oligosaccharides are raffinose, stachyose, and verbascose. Raffinose is nothing but sucrose with another glucose, another galactose molecule. So it is a trisaccharide of galactose, glucose, and fructose. What is shown with an asterisk there is the alpha linkage, and here you have an alpha 1,4 linkage. And raffinose, stachyose, and verbascose cannot be digested by the human body because we do not have the enzyme alpha 1,4 galactosidase, unlike we have beta 1,4 galactosidase. So therefore, raffinose, stachyose, and verbascose are undigested. The microflora in the gut ferment them. This causes bloating, discomfort, and flatulence. And I am sure most of you would have observed this when you have eaten chana dal, or you have eaten kidney beans, or you have eaten any type of the legumes. The legumes have, including soya bean, field bean, kidney bean, all the beans, the dry legumes are rich in raffinose, stachyose, and verbascose. So the only way of getting rid of raffinose, stachyose, and verbascose to prevent bloating and discomfort and flatulence is to cause germination. So during germination, and that is why you see sprouts are eaten, because in the sprouts, the raffinose and the stachyose are broken down, and you will find that the sprouts, if you eat green gram by itself, just soaked, it will not be sweet. However, when you eat sprouted green gram after 24 hours or 48 hours, you find that it is sweeter because it has been galactose has been broken down, and the raffinose has been broken down, and galactose has been released. So the sweetness of sprouts is due to the enzymatic cleavage of these trisaccharides. Trehalose is another very important disaccharide now in the food industry because it is used in several food as a humectant to keep the water, and also now it finds great use in sports foods. It is used in many sports drinks, and it is also known as the mushroom sugar. The reason it is used in sports drinks is it has the glucose alpha 1,1 glycosidic linkage, and it takes a long time to be broken down, and it gives you two molecules of glucose for energy. It has very low hygroscopicity. It has a high freezing point depression, and it can protect proteins, and so therefore it is also used to prevent drip loss in shrimp and other fish. Polysaccharides have more than 9 units of the sugar, and they can be polymers. And the three biologically important are starch, and cellulose, and all these are of the glucose. They differ only in their glycosidic bond. Starch has alpha 1,4 and alpha 1,6 linkages. It is found in plants, whereas its counterpart is glycogen in the animals. The polymers take up less room than glucose would, and so are more efficient for storage and compacting. So starch is made up of amylose and amylopectin. Amylose is an unbranched polymer which forms an alpha helix. You can see in the bottom panel, and it is linked by alpha 1,4 glycosidic bonds. On the other hand, amylopectin is more branched. It contains both alpha 1,4, what you see in the chains, long chains, and wherever you see a branch, there is an alpha 1,6 linkage. Almost every starch, whether it is tapioca starch, potato starch, or corn starch, they are mixtures of amylose and amylopectin. You do not get pure amylose and amylopectin. So normally the ratio is 65 to 85% of amylopectin and 15 to 35% of amylose. So depending on the ratio of amylose and amylopectin, the digestibility of the starches vary. So amylose is a straight chain, alpha 1,4. It improves the gut health. It improves glucose tolerance. Amylopectin is the branched form. It has about 3,000 to 6,000 glucose units. It is insoluble in water, and amylopectin gives you a red to violet color with iodine, whereas amylose will give you the deep blue color that we generally look and test for amylin. Amylopectin, the higher amount of amylopectin, decreases the gel strength and viscosity. And amylopectin is much easier to digest than amylose because you have the branches. So the enzyme can attack at the alpha 1,4 linkage and at the alpha 1,6 linkage. So what happens when starch gelatinization takes place? Those of you who have tried to dissolve starch in water will realize that starch will not dissolve in water at all. So you have to heat the water and stir continuously, and then you see that the starch becomes a little glassy. It starts thickening, it becomes glassy, and then it becomes very gluey. So the process of gelatinization is a complex process. It results in granular swelling, the crystallite melting, then the loss of bifringence, viscosity development, and finally the solubilized starch. So during the process of gelatinization, which requires the presence of water and heat, you will see that there is an intramolecular hydrogen bonding that occurs. And in gelatinization, you will see what is shown in red is the hydrogen bond formation between the OH of the sixth carbon, that is CH2OH, and the oxygen atom of the glucopyranoside residue. So all over you can see the intramolecular, that means within the molecules, you see the hydrogen bonding, and the gelatinization is due to the hydrogen bonding, that is the intramolecular hydrogen bonding within the molecules, that is between C2OH and the oxygen of the pyramid ring, and between two chains, that is between the CH2OH of the of A chain and with the OH of the B chain. So you have within the molecule, that is intramolecular, and you have between two chains, which is the intermolecular hydrogen bonding. Then in starch, you have seen there is a process known as retrogradation. It takes place when the gelatinized starch is allowed to cool and the glucose molecules will realign themselves. The most common part of retrogradation in starch is that in bread. You see, freshly baked bread is very soft, but however, after a day, it becomes a little hard, and we call this staling. So, staling is due to starch retrogradation. So, the retrogradation involves both the amylose and the amylopectin chains of the starch. The amylose molecules are unstable, and retrogradation results in an increase of turbidity and eventual precipitation. This occurs because the amylose will shrink, which is caused by a decrease in the kinetic energy and Brownian motion of polymer and water molecules. This shrinkage of the amylose results in the formation of both intermolecular hydrogen bonding between the hemiacetal oxygen atom and the adjacent OH6 and the O6 and OH2. So if you compare the hydrogen bonding of gelatinization, what you saw earlier, and now you see that there is much more hydrogen bonding, and because of this large amount of hydrogen bonding, starch retrogradation takes place. And this depends on the ratio of amylose to amylopectin. The structure of amylose and amylopectin in retrograding dictate the rate of the retrogradation, the temperature, the start concentration, and the presence of surfactants and salts. So you see, for those of us, the same maida should not be used for making bread and making biscuit. You have baking maida and you have the ordinary. So they are also modified. These starches are also modified for different activities. So quality defects due to retrogradation are, for example, staling of bread. You might be wondering why bread never remains soft. Loss of viscosity and precipitation in soups and sauces, and decreased storage stability. Starch can also be broken down into dextrins by the action of enzymes or by the action of acid, partial hydrolysis, and they are the main source of carbohydrate in proprietary preparations used as oral supplements for children. tube feeding. So you have dextrin for tube feeding, or it is known as liquid glucose. So liquid glucose is the mixture of maltose, glucose, and water. And these products are a means of giving carbohydrates in an easily assimilated form to patients who are seriously ill. The dextrin molecules are larger than sucrose or glucose, and they have a less osmotic effect. So, in the starch dextrinization, you see hydrolysis occurs and you get white dextrin. And when transglucosylation occurs, you get the yellow colored dextrin. I'm sorry, it is not there. So you have the white colored dextrin, which is due to complete hydrolysis, and the yellow colored dextrin is due to transglucosylation. So, what is the theory of the iodine test? So, iodine test is a very common test which we use to starch. So, when iodine is added, and you know in amylose you have an alpha helix, so rows of iodine atoms go into the core of the alpha helix and they form a dark blue complex. Whereas if you look at amylopectin, glycogen, and cellulose, you will see that there are no alpha helices. These are more of the straight chain. So therefore, iodine cannot go and sit in the core of the molecule. And so they show a purple or a brown color. And monosaccharides do not interact with iodine. Monosaccharides, disaccharides, and polysaccharides do not interact with iodine. So cellulose is the structural polysaccharide, and like starch, it is a polymer of glucose, but it is a polymer which has a beta 1,4 linkage. In glucose, you have the alpha 1,4 linkage, whereas in cellulose, you have the beta 1,4 linkage because it is a polymer of beta glucose, that is the anomer of alpha glucose. Notice highlighted, you see the OH group on top, which shows that it is the alpha, is a beta glucose. So cellulose, we cannot digest, the human body cannot digest cellulose. We do not have the enzymes. So therefore, it digestive tract as insoluble fiber. Some microbes use enzymes to digest cellulose. The enzyme cellulase is there. Does not have this, but many herbivores from cows to termites have symbiotic relationship with these microbes. So remember the difference between starch and cellulose. Starch is a polymer of alpha D glucose in alpha 1,4 linkage, whereas cellulose is a polymer of beta glucose with beta 1,4 linkage. Then we come to the dietary fiber, also known as the non-starch polysaccharides. This concept was made by Hipsley in 53, and then later dietary fiber was redefined as the skeletal remains of the plant cell that are resistant to digestion by enzymes of the human. So dietary fiber, it was suggested, should be measured as the non-starch polysaccharides in plant foods. So Englist proposed that a dietary fiber should be defined for the purpose of food labeling as non-starchy polysaccharides. So what are these non-starchy polysaccharides? The fiber is very good for health. It is the non-digestible polysaccharide. It provides no energy. The fiber can be classified into soluble fiber and insoluble fiber. In the soluble fiber, it forms the pectins, beta glucan, some gums, and mucilage. What is mucilage? Those of you who have eaten okra or bhindi, lady's finger. You know, when you cut it and you put it into water, you get that slimy liquid. That is known as mucilage. It is easily fermented by the intestinal bacteria into carbon dioxide, methane, and short chain fatty acids. Whereas the insoluble dietary fiber is the cellulose, the lignin, and some hemicellulose, which is not easily fermented. So, health benefits are there for both soluble and insoluble fiber. For soluble fiber, it slows down the gastric emptying. Beta glucans help to reduce the serum cholesterol. The soluble fiber also helps to normalize the blood glucose, and the soluble fiber helps to protect against colon cancer. In fact, for colon cancer, the pectin from orange peel pectin is given in large quantities to those patients with colon cancer. And the insoluble fibers such as cellulose, lignin, and hemicellulose actually give a lot of roughage and they relieve constipation. So that is why you are asked to eat green leafy vegetables. So in the green leafy vegetables, the cellulose will not be digested. And plants will contain both soluble and insoluble fiber in different ratios. So hemicelluloses, and pentose, they are made up of xylans, which clearly the name suggests that it will have xylose and with side chain of glucuronic acid and arabinose, mainly in wheat, found in wheat, rye, and barley. The galactomannans, the mannose backbone with galactose and glucose, found in legumes, and a very rich source of galactomannan is the fenugreek seeds or the methi seeds. So if you soak methi seeds, you will see that it absorbs a lot of water, and so it helps especially to absorb glucose. Then you have the xyloglucans, the name suggests is made up of xylose and glucose. So three different types of hemicellulose. The beta glucans found generally in oats. That is why now you find everybody eating oats either for breakfast or thing, because it is very well proven that the beta glucans of oats help to lower the serum cholesterol. So unlike cellulose, the glucose chains are branched and they have the beta 1,4 linkage. And as I said, they have become very popular because of the cholesterol reducing property. Then you have another polymer, which is inulin. It is a plant polysaccharide and it is a polymer of fructose. And it generally is found in seaweeds, and also it is found in chicory. Chicory is a root which is used to give chicory powder is added along with pure coffee powder to give body to the coffee. And this root contains large amounts of inulin, and it can be hydrolyzed when you think to give you fructose, which is sweeter. And if you look at the right hand side, it is a polymer of fructose, but the end residue is a glucose residue. So it has a glucose residue, and it, as I said, it is found in onion, garlic, banana, asparagus. But the richest source is uh, chicory as well as artichoke. Pectins found in all fruits and vegetables, especially the citrus fruits, and in the citrus rind, there are two types of pectins. The rhamnogalacturans, the name suggests it contains rhamnose with galacturonic acid, and you have the arabinogalactans, which contain arabinose and galactose. These pectins, not only are they very useful as the soluble fiber and as nutraceuticals for cancer patients, but they are also used as stabilizers, emulsifiers, and gelling agents in jam. Gums, these are water soluble viscous polysaccharides with very, very high sugar numbers from 10,000 to 30,000. It contains glucose, galactose, mannose, and they are extracted. One of the famous gums is guar gum. Guar gum is made from a legume, which is guar beans. I don't know what the Hindi name is, but those of you who know Tamil, it is Kottavarangai, and those of you who know Kannada, I am not, I've forgotten thing, but guar gum is very good, and the guar beans are very, very good and very bitter. So you have examples are gum arabic, which is an exudate from acacia tree, and then you have locust bean gum, which are galactomannans. Locust bean gum is one of the best gums because of the chemical nature of it. However, it is very expensive, and people are looking in the industry to get a gum similar to locust bean gum. So the properties of these gums will vary upon what sugars they contain. Then you have mucilages. As I said, the main mucilage, if you get, is from the lady's finger or bindi. They are characterized by having galacturonic acid. You can find it in seeds, roots, seaweeds, and algae. Examples of are alginic acid from the brown seaweeds, which contain mannuronic acid and guluronic acid. And you have carrageenan, which is a sulfated galactose polymer. These are all allowed, the gums, all these gums are allowed as food additives by FSSAI. Coming to the animal polymer, branched form of a glucose polymer. It is stored in the liver and muscle. It is stored for about 1 day in the liver, and it is constantly broken down. So if you are fasting, your liver glycogen will be broken down to give you energy, and it is broken down in response to the blood sugar. So when your blood sugar drops, glycogen will be broken down and glucose comes into the blood. And when the blood sugar goes up, then the extra glucose will be stored in the liver as glycogen. Your muscles also contain glycogen, and these are broken down by energy for the muscle. So you look at the glycogen molecule compared to the starch molecule, it is highly branched, and it has 1,6 and 1,4 branches. So this is to just a recap for you to compare the plant storage polysaccharide and the animal storage polysaccharide. Two types, the starch contains amylose and amylopectin, whereas animal has only one. Amylose is not branched, amylopectin is more branched, whereas glycogen, it is highly branched, very similar to amylopectin. Then you have chitin, which is a polysaccharide which is found in the exoskeleton of insects. It is a polymer made up of N-acetyl glucosamine, that is an amino sugar, and chitin is used in surgical threads, and it serves as a protection from water in the insects. So their exoskeleton is made. All the crustaceans, such as the shrimp, prawn, etc., their exoskeleton is made up of chitin. That means they are water insoluble. But chitin is now being used, it is broken down, and you get chitin oligosaccharides and the chitin biocides, which are known to have a lot of biological activity and are used as nutraceuticals. The ABO they contain the sugar molecules at the end. So you see type O is characterized by fucose, galactose, and N-glucosamine. The type A has fucose, galactose, N-acetyl galactosamine. They are all different. So you see they have the backbone of N-acetyl glucosamine, galactose, and fucose, but type B has a galactose residue, type A has an N-acetyl galactosamine residue, and type O has no residue. Then you have heteropolysaccharides, which are important of animal origin. You have the hyaluronic acid, which is a viscous polysaccharide, which is made up of N-acetyl glucosamine. This is found in the connective tissue and acts as an intercellular cement. You have chondroitin sulfate. The name itself suggests that it is a sulfated sugar, and it is composed of glycosaminoglycan. And then you have heparin, which is a mucopolysaccharide which contains long chains, as you see, of iduronic acid and D-glucuronic acid, and it is sulfate. So heparin is the anti-clotting factor in the blood. So all these are carbohydrates. It is good that you know that other than glucose, fructose, lactose, maltose, other polysaccharides or other saccharides are also there in the human body, which play a vital role in maintaining both structure and function. Now let's come to the reactions of the monosaccharides. So if you look at the monosaccharides, they have got either a aldo group or a keto group. And we know that the aldo group and the keto group are carbonyl compounds. So just like in organic chemistry, these carbonyls can also react. So the aldehyde of glucose can be reduced by using sodium borohydride to get glucitol. So the CH2 is reduced to CH2OH. In fructose, the CO is reduced to CHOH, and you get D-glucitol and D-mannitol. The reduction of these monosaccharides to sugar alcohols has applications in the food industry, in that these sugar alcohols, that is glucitol, mannitol, sorbitol, which are there, are all used as sweeteners because the caloric value is much less than that of sugar. So the sugar-free chewing gum, xylitol or sorbitol, all of these have, because it does not add to the calories. But these have got laxative effect. That means they cause a little bit of diarrhea. Those of you who have been chewing gum continuously would have experienced this. So the sugar alcohols found in nature are sorbitol, which is found in fruits, and it is used in the manufacture of soft drinks, chocolates, sweets, etc., because it is 60% as sweet as sucrose. Mannitol and dulcitol are alcohols of mannose and galactose. It is extracted from seaweed. Inositol is a cyclic alcohol which is present in many foods and in cereal brands, etc. Then, just like how the reduction of the carbonyl group took place, you have the oxidation. So the CHO of the sugar alcohol with the red arrow pointing to it can be oxidized to the acid, that is the COOH. So the sugar can become the aldonic acid. So carbohydrates that can be oxidized to aldonic acid are reducing sugars. The reducing sugars are oxidized to aldonic acid. So those of you who have done the test for monosaccharides would have done either the tolerance test or the silver mirror test, which is due to the oxidation of monosaccharides. Now, that was the oxidation of the carbonyl carbon, that is the aldehyde group. But the CH2OH, that is the secondary alcohol of it, also can get oxidized, and this gives what are known as the gluronic acid. So you have the aldonic acid and you have the gluronic acid, depending on which of the whether the carbonyl carbon or the secondary alcohol is oxidized. So, most of you are familiar with the Benedict's reagent. That is the reagent which is used when you add a saccharide to it or a reducing sugar. From the blue solution, when you heat it, what you get is a brick red color. So, how does this reaction occur? So, you see aldoses and ketoses in their open chain form will have either the aldehyde group or they will have the keto group. So Benedict's reagent contains copper sulfate or it has the blue Cu2 plus ions in it. So when added to that, the Cu2 plus ions are reduced to form the Cu or the cuprous oxide, which is the red color in it. And aldoses and ketoses in the open chain are oxidized by Benedict's reagent to form carboxylic acid and the red color. So sugars can thus be oxidized, are called as reducing sugars. So that is because the anomeric carbon is free. So wherever there is an anomeric carbon or a reducing end, they will react they will get oxidized to the aldonic acid and they will reduce the compound with them. So cellobiose and maltose are reducing sugars, lactose is a reducing sugar, but sucrose is not a reducing sugar. Not only do the structures show it, but if you take sugar and add Benedict's reagent and heat it, you will not see any color change. So several tests are there to test for carbohydrates. The first test is Molish test. So in the Molish test, this gives a rapid positive test and it tells you that a carbohydrate is present. So you can differentiate carbohydrates, lipids, and proteins. When you add Fehling solution and you boil it, it can distinguish between reducing and non-reducing sugars. Reducing sugars will give you a brick red color, whereas the non-reducing sugars will not react with Fehling solution. Benedict solution, also a different form similar to Fehling solution, contains the copper, the copper sulfate, along with the Rochelle salt or the tartrate. And again, it is used to distinguish between reducing and non-reducing sugars. So all monosaccharides, lactose and maltose will give the brick red color, whereas the disaccharides such as sucrose will not react with Benedict solution. Starch also will not. Barford's test distinguishes between reducing and non-sugar reducing also. Bayer's test will tell you it distinguishes between the hexoses and the pentoses. All the pentoses will give a green color with Bayer's reagent, whereas hexoses shows no color. Selivanov's test is used to distinguish between ketoses and aldoses. So fructose and sucrose will give a cherry red color with Selivanov's reagent, which generally contains resorcinol. So generally the estimation of sugars, the quantitative estimation of sugars can be done either by chemical, polarimetric or chromatographic methods. So the common method that is widely used is the Lane and Einan method. What is the principle of the method? How many of you have been using this method and have you thought of the principle? Here, the principle is very simple. It contains Fehling solution. Fehling solution is a mixture of copper sulfate and the alkaline Rochelle salt sodium potassium tartrate. So when this two solutions, that is Fehling solution A and Fehling solution B, are mixed, then copper hydroxide is formed. So the reducing sugars will reduce the cupric hydroxide to cuprous oxide, and what you get is the red color. And to this we add an oxidation reduction indicator, which is methylene blue, and that will detect the end point. So the test takes advantage of the ability of the aldehyde to reduce the Cu2 plus to Cu plus. Cu2 plus is blue in color, and Cu plus, that is the cuprous, is red in color, and it gives you an insoluble precipitate. So in order to do this titration, it is a very simple titration. The Fehling solutions A and B are mixed just before the titration, and they are heated so that the cuprous, the CuOH2 is formed, and then the sugar solution is added from the burette, and with continuous heating the titration is done. What is shown in the bottom is the overall reaction, that is the aldehyde reacts with two molecules of Cu2 plus to give you Cu2O, which is the rust red in color. So the blue color is that of copper sulfate, and the red color of that of the titration is due to cuprous oxide. So here is the steps in titration, and the titration must be completed in about a minute. So you mix Fehling solution A and B, that is copper sulfate solution, with the sodium potassium, alkaline sodium potassium tartrate solution, heated. Now you add the sugar solution from the burette, and when you see the reddish color not disappearing, then you add the indicator methylene blue. So just before the last few drops is added, this is what you see, a bluish color and the end point. So generally in this titration, the approximate volume, initially a titration is done to give you an approximate value of the glucose required to reduce the Fehling solution. And then in your second and third titration, you can find out the exact amount. And starch also is estimated in the same way, but here the starch will be broken down, or it will be hydrolyzed into the glucose and fructose. Both glucose and fructose are reducing sugars, and then you titrate the inversion or the hydrolysis product of starch against the Fehling solution. But one precaution that must be taken during the titration of starch is that before the starch is hydrolyzed, there should be no free sugar. So all the sugar must be completely washed off from the starch before it is hydrolyzed. Then the starch is hydrolyzed to glucose and fructose with dilute hydrochloric acid in a boiling water bath and titrated with Fehling solution, just as in the previous case. So with this, so here you can test for removal of sugar again by using the Molish reagent, that is the reagent. So you wash the starch continuously till you get a negative reading with the naphtha reagent. Then you can hydrolyze the starch and do it. So with this, I have covered the carbohydrates, what is the classification, the isomerism in carbohydrates, mutarotation, optical rotation, what are the important carbohydrates of the diet, disaccharides, polysaccharides, differences between the structure of starch, cellulose, and glycogen. What is dietary fiber? What are the constituents of dietary fiber? The importance of dietary fiber, the gums, the alginic acid, etc., and also how to analyze the carbohydrates.